sectioning, you would see this is a triad. You see a triad on each edge, right?
Yeah.
So there's one triad. There should be one there. Should be one there. Here, right there, there. So that outline right there, that's a lobule.
Okay.
Follow?
Yeah.
So are you recording this?
Yeah.
Let me make that lighter. Can you actually see it? Is that cool?
Yeah.
Okay, so this is the outline of the lobule in one of the four lobes of the liver. These are the portal triads, and that's going to consist of this right here, which is the hepatic portal vein, which leads into the sinusoids that then drain in, after detoxifying, drain into the central vein. They also are going to have a bile duct that collects the bile through canaliculi from each of the hepatocytes, and then you'd have a small arterial, which is the hepatic artery. Right, so let's just magnify that one. And this is a nice big one right over here. But it's got, you know, that weird stuff right there, that big, odd-looking material right there. So that's something that is a larger convoluted tubule that we should steer clear of. What I would find as the right structure to use here would be that trio of structures. Right here is the cuboidal-lined duct, bile duct. Here is the small arteriole, and this is the hepatic portal vein right there with blood vessels, blood cells inside of it. These would be white blood cells. I'm sure you have some anucleate red blood cells also if we were to look closer. And so, yeah, so there's the white blood cells in the lumen. This is the endothelium that lines it. This is the hepatic portal vein. So this would lead into the sinusoids, or the pathways that you see, you know, like that right there. There's one. Oh, it's the best. There we go, right there. So notice this hepatic portal vein goes into a pathway like this that leads towards the central vein, which is right over here in the center. And we'll find it in a moment, right? So, anyway, that's right there. That's the central vein, okay? So these sinusoids or pathways are leading to that, which it will then collect in the hepatic vein and go into the inferior vena cava and back to the heart. Right? So if we can find that triad again. Well, let me back off here. There's the central vein right there. There we go, right there. So the portal triad then that we're looking at here will have three structures. Right, so here we're looking at the hepatic portal vein. Then you have a small arterial, and then a cuboidal-lined. There's the cuboidal-lined hepatic duct, or sorry, bile duct. This is the hepatic artery, right? So and then here is the hepatic portal vein. So we would. This is formed, this structure right here is formed by the combination, if you want to grab me that model right there, the pancreas one, yeah. Because on this model, you can see, you know, we can trace the whole pathway. So if you notice right here, this is the splenic vein. Right there is the inferior mesenteric vein, and there's the superior mesenteric vein, all converging at this point, the hepatic portal vein. The horse, yeah. So this hepatic portal vein is what we bring all of the things that have been absorbed from in the digestive tract down here, you know, since it's the mesenteric arteries and veins, we're looking at the small intestine and large intestine's absorbed material. So if we bring that back into this hepatic portal system and into these sinusoid pathways here, the sinusoids contain Kupffer cells and run along through the hepatocytes adjacent to them. And as we saw right up here, right there, we have pathways that lead into that central vein here. So these are hepatocytes along the side. That right there is in the actual lumen, isn't it? So that's a Kupffer cell right there. And these would be the hepatocytes, both of which are helping with detoxification of the blood and materials that have just been absorbed from the three veins that lead into this hepatic portal pathway. Then as you detoxify and you send it forward into that central vein, it'll be then detoxified and deoxygenated and need to be sent to the lungs for oxygenation, at which point it'll come back from where it was oxygenated into this triad. I just missed it again. Down here, right? right? And there's a larger hepatic portal vein. Well, you can even see each and every one of the white blood cells within it. Right, so there's the neutrophils right there. That looks like an eosinophil down there. And you see the cytoplasm is pink staining right there. Right, so anyway, and these are red blood cells within it. So this hepatic portal vein is the beginning of the process of breakdown after absorption and utilization of the nutrients that have been brought in. Okay, and as it's sent through those sinusoids, you detoxify, you send it to the right atrium of the heart through the inferior vena cava from the hepatic portal or hepatic vein that is collecting all central vein materials. Then once it's oxygenated, you bring that blood back in through the celiac trunk and into the hepatic artery, and you provide all these cells what they need, especially the hepatocytes, the abundant cell in this slide, so that they can produce the bile that'll then move through the canaliculi and bile duct back to the common hepatic, join with the cystic, and form the common bile duct. Right? So it's a pathway that continues all the way through. If we had the large liver, I would do that with it too, if you wanted to. There it is right over there. You want to grab the large liver? Yeah. Okay, so this is the hepatic portal vein, which is going to be absorbing or bringing in the blood that has absorbed nutrients but hasn't been detoxified yet. So it's this inferior mesenteric, superior mesenteric, and splenic coming together here. And this is the liver. So in the liver is made up of four basic lobes. We have the caudate lobe, the left lobe, the right lobe, and the quadrate lobe. And those lobes are made up of lobules, right? And those lobules are microscopic. And so when we look at them, those lobules and the outline of them at least, like we see here, this is the outline here of a lobule, and so hundreds or thousands of lobules with its 6 sides and the portal triad surrounding it will have 3 structures within it that are going to be in the line of liver function. So if we look at the liver here, all of the nutrient-rich but non-detoxified blood will come in through this, the hepatic portal vein. That's going to then be spread out into the hepatic portal veins that branch from it, into the lobules that you see here, right? So, and these lobules then, the triad right there is the hepatic portal vein component. Through the sinusoids and Kupffer cells and hepatocytes, we detoxify towards the central vein, and the central vein then would collect the blood and send it back through, now that it's detoxified, into this, the renal vein, the hepatic vein, which then dumps in to the inferior vena cava, sends back to the heart for oxygenation in the lungs, and then it returns through this, the hepatic artery, oxygenated and detoxified. And then if you look here at the lobule, that small arteriole that we see right there is what's delivering blood to the hepatocytes to make bile. The bile itself will be created within the lobule, sent through canaliculi, and collect in the left and right common hepatic, or left and right hepatic ducts. That will combine to the common hepatic, which will join with the cystic duct from the gallbladder and form this common bile duct that we see here, joining and sending bile in through the hepatopancreatic ampulla with the main pancreatic duct to break down the food or emulsify the fats in the food that we've eaten. Study.